Hello and welcome everyone. Thank you for joining us today for our 10th webinar, organized by the Young Researcher Association of Institut Pasteur de Tunis in collaboration with Institut Pasteur de Tunis. I'm Khawla Ben Farhat, a postdoctoral researcher at Institut Pasteur and a member of the Young Researcher Association. And I will be the moderator today for today's webinar. So first, I'm going to introduce briefly our speaker today. So it's our pleasure and an honor to have with us Dr. Hamdi Mbarak, who's currently an assistant professor at the Department of Biological Psychology of VU University in Amsterdam, as well as a scientific partnership manager on the Qatar Genome program launched by Qatar Foundation. His research interests concern genetic and epidemiological analysis of fertility-related traits and reproductive disorders. And he's especially interested in the genetics of twinning as a measure of fertility. In today's webinar entitled Understanding the Impact of SARS CoV-2 on Host Genomes, he will give us an overview about the COVID-19 Host Genetics Initiative, which brings together the human genetics community to generate, share, and analyze data to learn genetic determinants of COVID-19 susceptibility, severity, and outcomes. And he will also share with us some of the obtained results thanks to this international collaborative effort. So, as usual, we invite all the participants to be interactive and to send their question. And with no further delay, thank you very much, Dr. Mbarak, for accepting our invitation, and the floor is yours.
Good morning, Sabah Khair. Thank you, Dr. Khawda, for the nice introduction. First, I would like to thank l'Association des jeunes chercheurs de l'Institut Pasteur de Tunis for inviting me. It is always an honor and a pleasure to talk in front of you virtually this time, unfortunately. So as you kindly introduced in this talk, I will be presenting the latest work we have been doing as part of the COVID-19 Host Genetics Initiative, which is a worldwide effort to understand the role that our genetics plays in COVID-19 infection and severity. So let me go quickly through the outline of my presentation. I will first start by a bit of background on COVID-19. Then I will introduce the Host Genetics Initiatives. What are the phenotypes that we are studying here? I will also guide you through some of the results and the follow-up analysis that we are doing at the moment. I will finish my talk by presenting also other projects that are not in the core business of HGI, but they are closely related, and also some other initiatives that are going around. Okay, so as you know all, we as a scientist think that genetic susceptibility can play a role in explaining differences in individual disease susceptibility, severity, and prognosis, which is the case here for COVID-19, as we observed a large discrepancies in symptoms and disease severity between individuals. So by identifying these genetics variants linked to severity and outcomes of the disease, we could help in many aspects, like, for example, in finding the right therapeutics, in drug repositioning, or importantly also, since it is a pandemic and it's a novel virus, in identifying people at unusually high risk or low risk for developing such a disease. And this will also contribute to the global knowledge of the biology of SARS CoV-2 infection and disease. So as I said, since it's a pandemic, so the effort should be here worldwide effort, not a single country or a single lab or a single study who can do this, but it should be a joint effort of everybody. And we can do this, we can study this disease via two approach or two types of studies. The first one is retrospective studies, where we look at the biobanks and with existing genotypes and with existing connection to the healthcare system via the EMR or the Cerner system, which will get us real-time updates on the infection status and severity of the participants. The second type of studies is new studies. These are prospective studies, which are started immediately after this pandemic, around maybe January, February, or some cohorts started even a bit late in March or April. So this cohort started by consenting people, patients hospitalized, and to get their blood samples or biological samples, and to do a rapid genotyping or sequencing of the samples in order to participate in this analysis. So as I said, in late March, we launched this initiative. It was initiative by colleagues from the Broad Institute, MIT, Harvard, and Stanford, led by Mark Daly in collaboration with the Institute for Molecular Medicine in Finland. We launched this COVID host genetics initiatives in order to bring together the human genetics community to generate, share, and analyze the data, to learn the genetic determinant of COVID-19 susceptibility, severity, and outcomes. There is a paper which was published by the European Journal of Human Genetics introducing this initiative and the work we are doing in this consortium, and how you can participate and how you can contribute to this consortium even if you don't have data. So this initiative has three main goals. First one is to provide an environment to foster the sharing of resources to facilitate COVID-19 host genetics research. We are doing this by sharing protocols, questionnaires, IRBs. So because, as I mentioned before, there are some studies who just started now their prospective cohorts, and they do need help from others who might have protocols, questionnaires, and IRBs to use as a model. The second goal is to organize analytical activities across studies to identify genetic determinants of, as I said, susceptibility and severities. So the first is to prepare the environment. Second is to share these analytical activities by sharing analysis plan, by following protocols for the analysis plan. And the third goal is to provide a platform to share the results for such activities. So once we have the results, we need to share it with the community as soon as possible. And by having this consortia and by the help that many are providing to this consortia, we can have a platform where we share the activities, we share the data in maybe individual level data or summary statistics. And I will come back to that a bit later. So, but overall, this is a benefit to the broader scientific community. So we are not looking at one community geneticist, for example. It's for the broader scientific community. This initiative has received a lot of media attention, trying to find an answer to this disease using genetics. As you can see, it made the news in Science and magazine and some newspaper as well. So in these slides, I will present these initiatives in numbers. So for example, up to last week, this initiative has over 1,100 members. They visited more than nearly 70,000 unique users on the website. We have 200 studies that are registered in the website from 51 countries. There are 20 contributing studies among these 201 that actually participated or contributed with data in the latest round of the analysis, which I will present in the next slides. And the scientists, the community are also using a Slack channel. This is a quite famous platform for messaging between researchers and between scientists to exchange rapidly ideas and to discuss. It's like a chat forum for science. And you can see on the bottom right side of this slide that these are the list of countries that have shared the results in the latest round. And you can see that UK is doing very well with 4 contributing studies, followed by several European countries. It is also very nice to see studies from countries that are usually underrepresented in genetic research, including, for example, Qatar, Brazil, and Korea. You can help, as I said, if you don't have the data, you can help the community by joining one of the Slack channels and participated in the analysis and the writing of the analysis plan, in analyzing the data and doing some follow-up analysis. So how you can participate? Imagine you have a data, you have collected some, you have a prospective or retrospective cohorts, and you want to participate in this global effort. So how you can do this? There are two options. The first option is to share individual level genotype or sequence data. This is the way that is most preferable, but it is not always the case because of some restriction in each country. So for example, you can, once you submit your data, it will go through one of the European Archive for genome data. It's called EGA or the ANVIL by NIH, depending on your location. And this data will be, of course, very well protected and secured. These two environments for sharing the data, individual level data, you will do your analysis, or the analysis will be done, data harmonization, QC, and analysis will be done within this platform. And then the PI of each study will have the authority to allow other people or other researchers to access this individual level data. So it's not like you submitted the data and it's publicly available, especially raw data, it's publicly available to everyone. No, this is very well protected, secured, and people who need to access raw data, they need to apply and delete PI, who is responsible for giving this access. The second way to participate is to share summary statistics level, and this is what mostly many studies are doing, including my studies. So what we have done is the consortium shared a harmonized analysis plan. We followed this analysis plan. We submitted the data to a central place, and then there are analysts who are analyzing this data in meta-analysis fashion. And then the results are publicly available or immediately on the website of the initiative. So now comes the question of, I want to participate, but I don't have the budget or the resources to collect samples or to genotype or sequence these samples. Fortunately, the initiatives have many partners offering to do a free or discounting genotyping or even whole genome or whole exome sequencing. And these are several parties, as I said, interested to offer such contribution and to help other studies to participate. For example, there is the Erasmus in Netherlands who are offering to do a free genotyping using the GSA platform, the global screening platform from Illumina. They offer to do free genotyping for your cohorts. For your cases, if you have collected COVID-19 samples, you can send them to Erasmus or to Finland. They will do free genotyping for the cases, and they will offer you even a discounted genotyping for your control. So you can do your whole cohort for a very, very cheap price. There are also other famous biotech like Regeneron or Illumina or Thermo Fisher and many others you can see on the slide, who are offering also to help you in genotyping or sequencing your samples. But if you opt to use whole genome sequencing or whole genotyping for free, then you are asked to deposit your data in this European Genome Archive for the use by the community. And as I said, the access is very well controlled. So you can find all this information if you go to the website of the Host Genetics Initiatives, and you will find information about the analysis plan, about the phenotypes we use it, about data sharing, about these partners offering genotyping. And all details also are in this website. So next, it comes to the important part, which is the phenotype definitions. So in this slides, we summarized here the 4 types of analysis that we are doing, the case control analysis. As you can see, analysis A is about very severe respiratory confirmed COVID-19, and the controls can be can be chosen from any of the blue boxes in the middle. They can be laboratory confirmed patients. They can be everybody that is not a case, if, for example, control from population. They can be tested negative for COVID-19 and many other conditions. The cases, however, so analysis A is for very severe respiratory confirmed cases, and you can see the conditions and how to apply this, how to define your cases in the orange boxes. Analysis B is about hospitalized lab confirmed COVID-19. Similar, so you can use B1, V2, or B1 or B2 for depending on your selection of the controls. Analysis C is about partial susceptibility. Here we are looking at anyone who is tested positive, independent of the status of hospitalized or not. And the controls also can be from the population or laboratory confirmed negative controls. Analysis D is more into this to give more room for studies who don't have yet a clear phenotype. So here is about predicted symptoms. So predicted based on your symptoms, if we can predict if you can get COVID-19, or also based on self-reported questionnaires. So we used here a simple set of covariates, which includes age squared, sex, and age multiplied by 6 in all the analysis above, and this to consider to some extent the difference between younger and older. So, for example, the older are more susceptible to having other comorbidities. Okay, so here I start these slides by the first meta-analysis, which was available, I think, was around late April, so 1 month after we started our initiatives. And as you can see here, these results are still underpowered. We have at that time about 1,000 cases and over 600,000 controls. We used a simple analysis, COVID-19 diagnostic cases versus controls, and nothing is significant there. So the beauty of this consortium is that immediately after people share their results, like few days, a couple of days, we do the analysis centrally, the meta-analysis, and the data are immediately shared, publicly shared. So this should motivate groups to share their data for the community. So now the exciting part about the results. These are the numbers of cases across the different analyses. And if you remember, we have A1, A1, A2, B1, B2, C1, and so on. And here I kept this table only the cohorts with a larger number of samples, more than 50 cases. So I didn't include controls in this slide. On the top of the slides, you see a reminder of each category: severe hospitalized versus population control, hospitalized versus non-hospitalized, and so on. So as you notice, so the numbers are still too small, and we need to increase that. We have also a very small number of severe hospitalized cases. In total, we have 536 patients that have assisted respiratory support, and we start to see good numbers for the analysis B2, hospitalized COVID positive versus population control, and COVID positive versus population controls, nearly 7,000 cases. So these are still a little bit dirty phenotypes, but we hope with the time in the next rounds, more studies will join us and will increase the numbers for especially the severe hospitalized COVID-19. I highlighted here two of the studies that took part in this analysis, in this round 3, which are the Qatar Genome Project, which I did the analysis for about 13,000 whole genome sequence. So we took the approach to analyze the common variants generated by whole genome sequence approach, and also the study that I used to analyze the Netherlands twin register. Okay, so let's look a bit at the results of this analysis, of the round 3 of the analysis, which were available 29th of June. And if you start with the severe COVID hospitalized cases versus population control, we don't see anything significant. So please focus on the top red line, which represents the threshold for statistically significant variance. We don't have anything significant when we look at hospitalized COVID positive versus non-hospitalized COVID, as well as COVID positive versus COVID negative. We also don't see any significant results when we look at predicted COVID, these based on symptoms that are based on a model that was published to use symptoms to predict future COVID. Where we see significant and positive results in the analysis that compare hospitalized COVID positive versus population control, as well as COVID positive versus population control. And as you can see on the two left bottom of the Manhattan plots, when we compare these results, we found a peak here in hospitalized COVID positive versus control. This peak is located in chromosome 3. And if you notice that the peak is smaller, despite in the COVID positive versus population control, is smaller in terms of significance comparing to the above picture, despite the larger sample size of the COVID positive, where we have about 7,000 cases. So I will explain to you why in the next slides. So as you can notice also, the last two analyses are the largest analysis, and here stressing here that the sample size is really key in order to start see signals. Okay, so let's have a look a bit at the results of this analysis. And if we start with the severe COVID hospitalized cases versus population control, we don't see, the slide. So this is the peak on chromosome 3 that observed among different studies. The picture represents the forest plot, the effect size of this top variance within across the cohort. And this, you can see that the largest effect size is for the hostage study. This is a study that was reported a few weeks ago in New England Journal of Medicine. They found the same chromosome 3 hit when they analyzed severe respiratory failure cases from Italy and Spain. But I will come to that study a bit later. And so what we notice also that the effect size is consistent across the different studies. The signal is also there when we compare the hospitalized COVID versus non-hospitalized COVID. This is a more clean analysis because here both are COVID tested positive, and these are severe versus mild cases. However, as you can see, the confidence interval is large and does not allow to estimate precisely the effect size. It is interesting to see that it is still significant, although. When the effect size get attenuated, when we look at all COVID versus population control, and as you can see, this includes studies that are that have also in their cohort mild or moderate infection. So that's why the signal is a bit attenuated. And finally, we don't see any signals when we look at predicted positive cases. I think we can make a conclusion from this slides that this signal is really about severity and not about susceptibility. And it's also a very robust signal. So or Ancestry DNA, it's a famous DTC, direct-to-consumer company in US, which it's like kind of 23andMe. It's also about the same, but they are focusing more on Ancestry, and they started recently to send questionnaires and surveys to their cohorts. It's quite a huge cohort. They have over 500,000 participants. So we have been trying to see if the signals we have seen are replicated in these cohorts, and they started collecting detailed consumer survey, as I said, of swab tests, hospitalization, and other clinical data. And their first data release was available last week, and they looked at hospitalization versus not hospitalized positive, and second analysis, positive versus negative tests. And here are the results for our top candidate on chromosome 3. When we look at the first analysis, hospitalization versus non-hospitalization, we found that it is marginally significant. And for the second type of analysis, positive versus negative, it's not yet significant. So now this chromosome 3 hit, and which I report in the Manhattan plot here, it is very clear and robust signal, and I think it's worth that we work now as a community to try to follow up this signal up. We have two ways to go. One is to perform in silico and functional annotation of this hit, because this hit is a spanning cluster of 6 genes. So it's not only one gene, but it's a cluster of genes which might have all relevant function to COVID-19. And I will come back to that a bit later. So, and another way is to look at the impact on clinical measures among the hospitalized patient. So there are here many opportunities to different researchers to take leadership and to organize group around these research questions. And I believe researchers at Pasteur or at Pasteur Tunis or at universities or in CBS, FACS, or in Monastir, SUS, or in researchers in Tunisia in general are able to do analyze such data and to maybe perform take this top variants and do a quick TACMAN and quick association studies or a follow-up and expression data and the Tunisian patient. That will be very great. So in these slides, some of the in silico follow-up analysis that are now currently ongoing. Different groups signed, as I say, to do such in silico analysis, and I will present very quickly one or two examples of these analysis. So there are some groups interested in heritability, correlations, to do gene-based tests, fine mapping, to use expression data, to run a TWAS, or using the EMR data to run a FWAS, also to look at the correlation or the causation of the top variants, and so many other kind of analyses that are state-of-the-art analysis for genomic nowadays. So this is the initial report I told you about that was published a few weeks ago in the New England Journal of Medicine, where they analyzed severe COVID-19 cases with respiratory failure among cohort from Italy and Spain, with a total number of cases about 1,800. So the author found that frequency of the risk allele of the lead variants at chromosome 3 is higher among patients with respiratory failure than controls. And they also found that higher among patients with mechanical ventilation than with oxygen supplementation. So, however, patients with this respiratory failure correspond to a minority of all patients. So we know by now that most of the cases, COVID-19 cases, have a mild or asymptomatic, and only a tiny percentage develop severe symptoms and need therefore to be admitted at the ICU units. So the question remains if these variants are predisposing to infection or they predispose to a more severe form of the disease given the infection. They also found another signal which is located in chromosome 9 in the ABO locus, which is the blood type, but these signals have the results of these signals has been inconclusive, because we knew maybe a month ago or 2 months ago there is a Chinese study reporting that blood type may be if you have a blood type O, you are more protective. If you have the blood type A, you are more prone to risk. And and in this study, they found the signals, but it is weak. And when we look in our data for the host Gentex initiatives, I don't show the slides here for the sake of time, but we have seen inconsistency, and that the signals also is very heterogeneous and not replicated in the host Gentex initiatives. So that means that this signal maybe is not straightforward, and we should focus our effort first on the chromosome 3 signals. And this is what we have been doing. So maybe 48 hours after we put this results publicly available, groups in the VIEW in my university started to look at the gene-based test. They took the summary statistics and they ran a MAGMA gene-based association testing, looking at not only at single variants, but at the variants within the gene as a whole. And you can see the list of the genes that are spanning these regions, and you see by phenotype the number of genes that are statistically significant. And there are some very good candidates here, like the LZTFL1 gene. It is significantly associated with 23 immunological phenotypes, including macrophage inflammatory protein 1, monocytes, basophil counts, and there are 121 out of 296 genes that are associated with immunological phenotype. And at the bottom, the pathway analysis showed that for analysis B2 and C2, the one where we found chromosome 3, there are a pathway which is linked to complement, and another pathway which is linked to amyloid beta clearance by cellular catabolic process, and also immune effector process. So this is a kind of data or follow-up analysis that we can do quickly to get insights on the meaning of this chromosome 3 with regards to the severity for COVID-19. Another gene which also very interesting is the CXCR6 gene, which is regulates the specific location of lung resident memory CD8 T cells throughout the sustained immune response to the airway pathogen, including influenza viruses. And also other genes, or CCR9 is also the family of CCR5, the gene that was associated with HIV. So there are a lot of potential to study these genes to try to find what are functional consequences of this signal. Another kind of follow-up is to fine map this region, because, as I mentioned, it contains a cluster of 6 genes, as you can see on the lower panel here, and we don't know which is the causal variant, which is the causal gene. So the first thing is we need to do is to identify the causal variants from marginal association, because the cluster contains a lot of variants. So we need to know which variants are these first variants, are they independent and not, and are they linked to one gene or several genes? So a colleague from the Broad Institute used a statistical tool to fine map these regions. to try to find a single causal variance or a shared causal effect across all studies. So he found that 16 out of 20 variants in the 95 and 99% of credible sets, they are in tight LD in every population of GNOME ID, and suggesting that it might be challenging to find maps statistically. So since these studies include cohorts from different ancestry and ethnicities, so I think now the next step is to look at the individual studies and to look at carefully at the signal and try to find up within the study before doing find up on the level of summary statistics data. Next is the supported project by the initiatives, and these are analysts that are not part of the core analysis of the COVID-19 host genetics initiatives, but the initiatives are supporting them by providing the platform, by providing the data to do research. And here, for example, the clonal hematopoiesis group, they are quite active on the Slack channel with this tag. If you want to go to Slack channel, you can have access to this tag. They organize bi-weekly calls. They have quite well organized analysis plan, and they are looking for more studies with GWAS data on copy number somatic events associated with COVID-19, or on whole exome or the whole genome data to contribute to discover clonal hematopoiesis and intermediate potential chip data. Another project is the acute respiratory distress syndrome, and this is independent of COVID and maybe sometime pre-COVID period. So they are doing GWAS in BioFew and UK Biobank, which is done, and now they are replicating in other cohorts, and the results will be available by late July. Another group is also focusing on vascular complications of COVID-19, and the first round of genotyping is happening late July. Okay, so I will quickly finish with these slides on the major the chromosome hit variants that we found. There is a study which looked at the data from the New England Journal of Medicine and also on the data from the HGI, and they found that this variance is inherited from the Neanderthals. And these are just preliminary results, which should be taken carefully. They are on preprint server and not yet reviewed, but nevertheless, it was on the news last week, and a lot of interest is for such kind of data. The other initiative I want to talk about briefly is the COVID human genetics effort. It's international consortia aiming at the monogenic inborn errors for immunity, rare or common variants underlying the severity of COVID-19. They are looking also at monogenic inborn errors of immunity, making individuals resistant to infection by SARS CoV-2. So the main recruitment criteria are patient or adult patients or children without below 50 years old, without any major comorbidities, and with severe complications. So if you are interested or know someone who is interested, you can have a contact Casanova, who is the lead PI of this consortia. And finally, I want to introduce also a white paper that was out a couple of days ago from the International Common Disease Alliance. It's a recommendation and white paper. Very briefly, this is a scientific forum comprising international stakeholders across academia, medicine, biopharma companies, tech companies, and biomedical funders. The idea is to share how to best accelerate progress from maps to mechanism to medicine. So because now we have discovered about 70,000 robust genetics association with common disease, now the next step is to know how we can translate this variance from the bench to the bed. And in this white paper, which I really recommend you to read, it's publicly available. They provided a recommendation on several aspects, like bold scientific projects, which can take 5 to 10 years, on foundation of genetic resources, on computational tools needed to analyze such data, and on new research and training. So we've come to the end of my talk. Thank you very much for, again, inviting me, for listening, and I'm happy to take any question.
Thank you, Dr. Mbarek, for this interesting talk. So, and thank you for giving us also some hope that now it's the scientists and the researcher are more collaborating, and it's a good thing because it might help us to win this battle against COVID-19 or any other viruses in the future, hopefully. So we are waiting for a question, but I will start with something. So, I mean, when I saw the country that were participating in this initiative, we didn't see Tunisia. So, but especially with the situation here, and we are lucky enough to not have a lot of death in our country. So of course there is the preventive approach, but maybe there is also the genetic factor here, and it would be like an interesting core to explore. So do you think there is some experts in Tunisia that are going to come and work in this initiative, or there isn't really?
I hope so. I really hope so. I really hope so. And I'm trying to push and to help my colleagues at Pasteur or in other places in Tunisia to try to joint effort together. And as you can see, this effort would have never been possible if the whole community are not joining their efforts. So we should work together to try to collect samples, and this is a good timing, I think, that we change our mind and to try to collaborate closely on such pandemic. Coming back to your question, yeah, indeed, Tunisia is quite interesting case study. So we need to look. Fortunately, we don't have a lot of death, and we don't have a lot of large number of COVID positive. But it will be really interesting to see whether we are protected by our genetic background, or there is any other thing that we might, maybe vaccination. A lot of things has been said in the news about vaccination. So we cannot confirm. We need to do research. We need to study, and this is the way how to move forward, is to perform research.
Okay. So we have a question from Céline Boabit from Pasteur Institute. So she would like to know if there is a communication plan to share these results with the public.
Yeah, indeed. Hi, Céline. Indeed, this results, if you go to the website of the initiative, are publicly available. So what we do is we every time we have enough sample size, we run a round of we freeze the data and we run a meta-analysis. And immediately after the meta-analysis is done, the results are publicly available. Anyone can now go to the website, download the results. He can also download the follow-up analysis and the slides, because we organize every month, we organize a call to update the contributing studies. So you can go now and download this data. And as I said, I really encourage you and encourage researchers, young researchers, to download this data and to play with it. To maybe they have nice ideas, some innovative idea. Maybe they will look up at these variants. They don't need to do a whole genome or whole genotypes or sequence data. They can take the top variants and look at it in their expression data, and maybe in some animal models quickly in zebrafish. So you don't need to have a lot of resources. You just need to have a minimum of resources data that you can apply to and analyze your data. So please go and check the website, and if you need any help, don't hesitate to ask me. I'll be glad to help you.
Okay, thank you. So this is And a general question based on what you experienced during this new, let's say, adventure for this initiative. Do you think that now in research we are ready to move to collaborate more and go far from the competition that we were used to, or is it really specific to the COVID-19 subject, since it's like the topic of this day and really important for everything, or is it going to be something more worldwide spread for other topics?
I mean, we should seize this opportunity. It's really the time to collaborate. Such effort, such initiative it would never have been possible if not the whole community are joining their efforts. It is amazing what has been achieved in only 3 months. Such project, and I have been involved during my postdoc and during my past experience. I have been involved in many large consortia, and usually to assemble such a cohort and to work together with many contributing studies around the world, you need, let's say, 3 to 4 years to come up with a paper, with a publication, or with something meaningful. This initiative has been, we started to generate the data within a month. So if there is no collaborative effort, if we forgot about competition, then as you can see in my slide, so nothing is written in stone other than the obvious. We must all act together and with no personal gain or ownership of results, just rapid and immediate dissemination of the maximum possible data and information that can be responsibly released and shared. So I think it's the time now, and given we are a small country, so we should join effort, work all together,
Hopefully.
and help the community, because this is a pandemic. Everyone is affected. It's not only one country or one study, everybody.
Okay, thank you. So we have another question from Sonia Matouk from Institut Pasteur. So is there any association between the virus genetic variant and symptoms developed on people?
Yeah, this is an interesting question, Sonia. I think the next step is to try to link the sequence, the viral sequence, to the host genome sequence. And there are studies who are now looking at this, because in the same time, there are a lot of efforts also to sequence the viral genome. And I know at Pasteur, the virology lab, or at Charnikor, or other places, maybe in Tunisia, they are also trying to sequence, or they did already some sequence. So it will be very interesting to try to link this to the viral genome to the host and see if, depending on the viral load, if the person can develop severe symptoms or not. So this is a very good question, and I hope the next step is to combine viral and host genome to study the consequences of this infection.
Okay, so
and similarly, also, sorry, I want to add. Similarly, we need also to join efforts to sequence the viral genome, because it will give us a lot of information on the spread of the virus in the country, on the community that are more affected. And we should also share this data and make it publicly available to know what strains of the virus are in the country, and what are the introductions that we have. And there are also large databases that assemble such kind of data. And I'm sure there are consortium who are doing the same thing, but for the viral genome.
Thank you. So I think that's it for the questions. So if you have anything that you want to add or final comment for us?
So thank you very much again for inviting me. If maybe many people will watch this offline, I'm happy to answer questions. I'm happy to help you to get access to the data, although it's publicly available, but maybe you are lost in the number of analyses, or you want a specific question, or you want to extract one or two variants. I can do that for you. I wish next round I will be able to help you guys to analyze Tunisian samples, to participate, because so far among the countries that are contributing, there are only Qatar from Arabic world that are participating, but we are very much keen to see more diversity in this consortium, and a lot of partners are willing to help to genotype or sequence samples. So please get in touch.
Yeah, I think we have some more questions that are popping now. So another question from Sonia Matuk. After gathering all of these results, what is the next step? How will these results will be used?
Yeah, very good question also. So this is the first signal. We start after round 3, and after ramping up our sample size, we start to see the first signal. Chromosome 3 is quite a robust signal. As I mentioned, it's linked to severity. We need to do a lot of follow-up to delineate the functional consequence of the signals, but also we need to work to increase our sample size, because there might be other signals that are associated with COVID-19. So a lot of things are happening now, and yeah, so there are studies who are working on functional analysis. So it will be interesting to see how we can help the clinicians to protect or to avoid that we have this severity in symptoms, and to protect people that are at higher risk.
Okay. Next question from Mariam Houas, also from Institut Pasteur. So can the environment, diet, vaccination influence the susceptibility to the virus?
It is certainly not only genetics. There are certainly some environmental factors, some diets, maybe vaccination. So we need to study this to know. We cannot. We need to do research, and there are groups who are interested in this. So we need to study it very carefully and to participate in these questions. Among the initiatives, there are studies who are interested in vaccination, who are interested in microbiome, for example. So if you have some ideas, or if you want to participate, you can always join the Slack channel and subscribe to the website, and you will get update about what is going on.
Okay. Thank you, Dr. Mbarak, for your talk, and for all these answers and for this interaction. And now I would want to thank everyone for attending today's webinar. We hope to see you in the next one that will be held on July 20, and covering the topic of technology transfer and valorization. So thank you again, everyone, and take care.
Thank you. Bye-bye.